faithfully convey value and interpret great books from around the world. Hello, this is Sally's Book Talk. Today, the book we're discussing is called The Neanderthals. In 1856, about 10 kilometers east of Dusseldorf, Germany, in a place called the Neander Valley, workers were clearing a small cave in a quarry when they unexpectedly discovered a skull and some other bones. And at first, these bones were thought to be the remains of a bear. But after a few years of examination, scientists discovered that the morphology of these bones was very similar to that of modern humans. They were fossils of ancient humans. And because they were discovered in the Neander Valley, this ancient human species was named Neanderthals. As archaeological excavations progressed, similar human fossils were discovered in multiple European countries such as the UK, France, Belgium, and even in West Asia and Central Asia. Scientists collectively called them Neanderthals, and they were once considered the direct ancestors of modern humans. But later research overturned this view. So, who exactly were the Neanderthals? What was their relationship with modern humans? Well, today the book we're discussing will start from the perspective of DNA research and gradually uncover the secrets of the Neanderthals. The author of this book, Svante Pääbo, is a Swedish evolutionary geneticist and the current director of the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. Writing Lights, he is also a member of the Royal Swedish Academy of Sciences and the Royal Society of London. And Pääbo is one of the pioneers in the field of paleogenics, dedicated to the study of ancient DNA sequencing of Neanderthals and a trailblazer in Neanderthal research. In 1997 and 2010, the research teams he led published two groundbreaking papers on Neanderthal DNA sequencing. In this book, Pääbo not only recounts the discovery and research of Neanderthals by combining his own experiences, but also shows how theories about human origins have been revolutionized step by step, right? So it's one thing that next, I will interpret the book for you by breaking it down into three questions. The main content of the book. First, why did we initially believe that Neanderthals were the ancestors of modern humans? Second, how has the development of DNA technology refreshed our understanding of Neanderthals? And third, what exactly is the relationship between Neanderthals and modern humans? All right, let's start with the first question of the book. Why? Why did we initially believe that Neanderthals were the ancestors of modern humans? In the earliest times, people believed humans were created by deities. For example, the book of Genesis in the Bible states that God created the earth and all life, including humans, about 13,000 years ago. Furthermore, these species were believed to never change. In the 1850s, Darwin proposed the theory of evolution, suggesting all things evolved gradually over billions of years, with humans evolving from apes. During the 19th century, numerous paleontological fossils discovered in Europe strongly challenged creationism. For instance, Archaeopteryx fossils date back over 100 million years, far exceeding the 13,000 years since Earth's divine creation. Our focus today, the Neanderthals, lived between 200,000 and 30,000 years ago. Based on studies of Neanderthals and other human fossils, scientists proposed the multi-regional origin theory, suggesting humans originated in multiple areas. Neanderthal research reveals subtle morphological differences compared to modern humans. For example, their skeletons were broader, and their occipital bone, that's the back of the skull, was more prominent, resembling a hair bun at the back of the head. Other physical characteristics closely resembled modern humans. Neanderthals used tools, played music, and cared meticulously for the sick. They conducted ritual ceremonies during important festivals. Neanderthals were once considered ancestors of modern humans, especially Europeans. By the 20th century discovery, archaeologists discovered human fossils worldwide, with the oldest and most primitive, closer to apes, found in Africa. For example, the Turkana boy fossil from Kenya lived 1.6 million years ago, while Lucy from Ethiopia dates back 3.2 million years, and Australopithecus, who began the evolutionary journey from ape to human. Compared to Lucy, Turkana Boy was closer to modern humans. Based on leg bone morphology, he could walk fully upright, freeing his hands, classified as Homo erectus. In the early 20th century, paleoanthropologists established this human evolutionary path based on fossil dating. Australopithecus evolved into Homo erectus. Homo erectus began leaving Africa over 1 million years ago, gradually spreading worldwide. For example, they migrated to East Asia and evolved into the well-known Yuan-Mu man and Peking man, becoming ancestors of modern East Asians. In Southeast Asia, they evolved into Java man, ancestors of Southeast Asians. The branch that migrated to West Asia and Europe became the Neanderthals. So, this theory of separate regional evolution is called multi-regional origin. Scientists proposed this theory based on both fossil chronology and morphological characteristics. The morphological classification method originated from Swedish naturalist Carl Linnaeus, who published Systema Naturae in the 1730s, establishing systematic biological taxonomy. Simply put, it classifies organisms based on physical characteristics. The smallest unit is the species. A species comprises organisms with highly similar morphology. For instance, humans vary in height, weight, and skin color, but share similar morphology, belonging to one species. Following Linnaeus' system, similar morphology implied both classification and kinship. However, organisms adapt morphologically to environmental changes. Thus, morphology-based kinship determination isn't always reliable. For example, Linnaeus classified whales as fish due to similar appearance. We now know whales are mammals unrelated to fish, having evolved similar traits for marine adaptation. Similarly, Australia's extinct thylacine, called Tasmanian tiger, had wolf-like build and tiger-like stripes. Scientists initially believed it was closely related to wolves or tigers. Further research showed distant relation to wolves or tigers, but close ties to other Australian marsupials like wombats and Tasmanian devils. Though sharing only the pouch feature, as Australia's apex predator, it evolved wolf or tiger-like morphology for hunting. This led some scientists to question the close kinship between Neanderthals and modern humans. Especially with deeper archaeological excavations in the 20th century, Homo sapiens fossils were discovered in the same geological layers as Neanderthals. Just how similar were they to modern humans? If a Homo sapiens corpse were placed in a modern morgue, the coroner might not notice anything unusual at all. Although they lived tens of thousands of years ago, they already looked like modern humans. So what was the relationship between Neanderthals and Homo sapiens? And who is the true ancestor of modern humans? The multi-regional origin theory was questioned, turning the once clear human evolutionary path back into a mystery. Solving this puzzle depended on advances in DNA research technology. Now let's explore the book's second question. How has DNA technology refreshed our understanding of Neanderthals? With Homo sapiens fossil discoveries, some scientists propose that our ancestors were actually Homo sapiens who left Africa just 100,000 years ago, replacing other Homo species, including Neanderthals, to become modern humans' ancestors. This theory is called the single origin theory. From its inception, the single origin theory engaged in fierce debate with the multi-regional theory. DNA sequencing technology ultimately resolved this conflict. In 1953, American scientist Watson and British scientist Crick first proposed DNA's double helix structure. By the late 1970s, research into DNA structure had deepened significantly. DNA molecules consist primarily of 4 nucleotides: adenine, thymine, guanine, and cytosine, abbreviated as ATGC. These form a single strand in specific sequences, then pair with complementary nucleotides on another strand to form base pairs. Yes, this creates the familiar double helix structure. The sequence of base pairs is the genetic code that determines biological traits and reveals kinship between species. By measuring DNA sequence similarity, we can determine the relationship between Neanderthals, and modern humans. However, intact DNA only exists in living organisms where enzyme systems constantly repair damage. Upon death, these enzymes cease functioning. The double helix structure within cells most rapidly break and decompose after an individual's death. So at that time, some scientists believed DNA couldn't possibly exist in fossils or remains. But the book's author, Payabo, believed that under suitable preservation conditions, some DNA fragments could survive. In the early 1980s, Payabo, then a PhD student, borrowed mummy skin and soft tissue samples from a Berlin museum and successfully extracted DNA. This marked humanity's first DNA extraction for mummies and proved ancient DNA research feasible. This contribution earned Payabo an opportunity at Cold Spring Harbor Laboratory in the U.S., beginning his postdoctoral career. Cold Spring Harbor Laboratory is revered as the holy land of life sciences and cradle of molecular biology, ranking first among the world's top 10 most influential research institutions. It was here he encountered PCR technology, crucial for ancient DNA research. PCR stands for polymerase chain reaction, invented by American chemist Kary Mullis in 1985. Simply put, it's a technique to amplify DNA in vitro, dramatically increasing tiny amounts. This is the same technology used in current viral nucleic acid testing. Because viral content in original samples may be minuscule and hard to detect, but PCR amplification of viral DNA or RNA enables easy detection. Similarly, for fossils, scarce residual DNA requires PCR amplification for sequencing, determining nucleotide sequences. However, the human nucleus contains 23 chromosome pairs with about 6.4 billion nucleotide pairs, making nuclear genetic code deciphering difficult. But mitochondrial DNA research offers a breakthrough. Mitochondria are capsule-shaped cellular components providing energy, containing their own independent DNA. This DNA, called mtDNA, is human mitochondrial DNA. mtDNA has two key features. First, its relatively small size, about 16,500 nucleotide pairs, makes sequencing manageable. Secondly, mitochondrial DNA in humans has a unique inheritance pattern. It's only passed down through females. If a woman has daughters among her descendants, she has the chance to pass on her mitochondrial DNA. If she doesn't have children or only has sons, her mitochondrial DNA lineage ends. By tracing back through our mitochondrial DNA, we can find our earliest female ancestors. Moreover, because mitochondrial DNA has a simpler structure, mutations are generally minor, so it doesn't change significantly over tens of thousands of years. In the 1980s, scientists had already sequenced mitochondrial DNA from thousands of modern humans worldwide. Through comparative studies of these mitochondrial DNAs, scientists found that differences between them are minimal, essentially similar. In 1987, scientists inferred that all humans today trace back to a common female ancestor who lived 100,000 to 200,000 years ago, known as mitochondrial Eve. So who is this common mother? Was she a Neanderthal or Homo sapiens who looks more like modern humans? This requires sequencing mitochondrial DNA from fossils. From the early 1990s, the book's author, Payabo, began sequencing mitochondrial DNA from Neanderthal fossils. In 1996, his team obtained a small piece of a Neanderthal's right upper arm bone from the Landesmuseum Museum in Bonn, Germany. It was through this bone that Payabo successfully extracted Neanderthal mitochondrial DNA and completed the sequencing. The results showed that Neanderthal mitochondrial DNA sequences differ significantly from modern humans, so they couldn't be our ancestors. Using the same method, scientists studied other early humans besides Homo sapiens and reached the same conclusion. Only Homo sapiens and modern humans have similar mitochondrial DNA. Homo sapiens lived in Africa for hundreds of thousands of years and began migrating out of Africa around 100,000 years ago, replacing other Homo species like Neanderthals, Yuanmu man, Peking man, and Java man, becoming our common ancestors. At this point, The single origin theory replaced the multi-regional theory and gained widespread acceptance in the scientific community. Our ancestors were Homo sapiens who lived in Africa 100,000 to 200,000 years ago. They migrated out of Africa around 100,000 years ago and, through prolonged struggles, gradually eliminated other Homo species like Neanderthals, becoming the dominant species on Earth. Hearing this, you might wonder, how exactly did Homo sapiens replace the Neanderthals? While competing for territory, didn't they spark any romantic connections, meeting and producing offspring? Are we completely unrelated to Neanderthals? You're not alone. Scientists share these questions too. Actually, mitochondrial DNA sequencing only proves Neanderthals aren't our direct ancestors, not that they had no relationship with our ancestors.
Possible scenarios. If Neanderthal Homo sapiens interbreeding produced only sons, or their lineage died out,
It wouldn't appear in modern mitochondrial DNA. So do modern humans actually share ancestry with Neanderthals? Now we explore the book's third question. To resolve this, we must examine nuclear DNA. Recall, human nuclear DNA contains about 6.4 billion nucleotides. Yes, half comes from each parent. Unlike mitochondrial DNA, nuclear DNA is inherited regardless of gender. If interbreeding occurred, comparing modern and Neanderthal nuclear DNA could reveal traces among billions of nucleotide pairs. In the early 20th century, the decade-long Human Genome Project launched, 1990, a collaboration between the US, UK, France, Germany, Japan, and China. By sequencing modern nuclear DNA, scientists mapped global genetic characteristics. After this, Paabo, then director of Germany's Max Planck Institute, mobilized resources to study Neanderthal nuclear DNA. But first, finding suitable fossils was challenging. DNA sequencing requires fragment assembly. DNA extraction, whether from modern or ancient samples, yields fragments due to strand breakage. Fragments aren't problematic. They can be reassembled. Every cell contains identical DNA, theoretically obtainable infinitely from living tissue. PCR amplifies cellular DNA repeatedly, while computer systems automate comparison and assembly. It's like having a book where each copy has some torn pages, but with enough original copies, you can piece together a complete book from the fragments. Close quote. Although Neanderthal fossils have been unearthed worldwide, most contain minimal residual DNA, making few suitable for research. Typically, only fossils with over 1% DNA content qualify for nuclear DNA studies.
to complete assembly, Palbo's team examined numerous fossil specimens with insufficient DNA. Most fossil DNA was severely degraded due to two factors. First, acidic environments and time caused devastating DNA damage.
Only Neanderthal fossils preserved in alkaline environments might retain significant DNA, but even in ideal conditions, excessively ancient fossils, say over 100,000 years old, retained virtually no usable DNA. Second, fossils face contamination immediately upon excavation. Natural contamination occurs when exposure to air introduces bacteria. Human-induced contamination includes using gelatin made from cattle bones during processing, risking bovine DNA pollution. In traditional archaeology, some researchers licked fossils to authenticate them, severely contaminating DNA. Thus, only recently excavated fossils from alkaline environments with minimal contamination are viable for nuclear DNA sequencing. However, obtaining such fossils requires luck. New finds aren't annual, and non-German countries may refuse access to Pabo's institute. Such an opportunity finally arrived in summer 2006. Pabo's team obtained several Neanderthal fossils from Croatia's Vindia cave. These 38,000-year-old fossils included 3 with exceptionally high DNA content, nearly 3%. With suitable specimens secured, extraction could begin. Nuclear DNA also requires rigorous contamination avoidance. If Neanderthal nuclear sequences mix with modern human or other species DNA, results become unreliable. Pabo was most concerned about contamination issues. Pabo and his team first established an ultra-clean laboratory and implemented strict operational protocols. For example, entering the lab requires special coats, shoes, gloves, masks, etc. Additionally, all chemical reagents must undergo rigorous testing before entering. Any DNA detection, especially human traces, triggers immediate destruction. Secondly, to avoid sample contamination, they prioritized extracting material from fossil interiors, a highly delicate process. Lab technicians use drills to penetrate fossils for internal sampling. This requires sterile conditions and extreme slowness, as drill friction could heat fossils and damage DNA. Third, Pabo's team improved PCR methods such as optimizing polymerase use during DNA replication. Researchers developed polymerases less sensitive to non-target DNA, e.g. bacterial, reducing contamination interference. Ultimately, their Neanderthal nuclear DNA sequencing findings, published in a 2009 scientific journal, concluded: first, Neanderthals did contribute to modern human DNA. Though not our direct ancestors, they interbred with Homo sapiens tens of thousands of years ago. Modern Africans show minimal nuclear DNA overlap with Neanderthals, indicating interbreeding occurred after Homo sapiens left Africa. Other modern populations share similar Neanderthal DNA levels. Europeans show about 2% similarity. Despite no Neanderthal fossils in East Asia, East Asians also share approximately 2% DNA similarity. Second, Neanderthals and Homo sapiens share a common ancestor. Further DNA analysis reveals shared origins with Homo sapiens and other Homo erectus groups like Yuan-Mu man, Peking man, and Java man. All diverged from chimpanzees around 6.5 million years ago, sharing a common ancestor around 800,000 years ago. Subsequently, they gradually began to diverge. About 400,000 to 300,000 years ago, Neanderthals joined Homo erectus, migrating out of Africa, settling in Europe and West Asia, while our Homo sapiens ancestors remained in Africa until migrating out around 100,000 years ago. Through prolonged struggles, they gradually replaced Neanderthals and other early humans. Now we know that while humans aren't direct descendants of Neanderthals, we share an undeniable connection with them. Now someone might ask, if Neanderthals didn't live in East Asia, why are we related to them? The discovery and study of Denisovan fossils provide the answer. In 2008, archaeologists discovered ancient human fossils in the Altai Mountains bordering Russia and China's Xinjiang, later identified as a young girl's finger bone. Named Denisovans, research shows they were close relatives of Neanderthals who diverged after leaving Africa. After migrating from Africa, Denisovans traveled eastward to East Asia, with fossil remains found across China's Tibetan Plateau. After leaving Africa, Homo sapiens interbred not only with Neanderthals, but also with Denisovans during eastward expansion. The Neanderthal-like genes in modern Asians actually came from Denisovans. Neanderthals, close relatives. Studies suggest some distinctive East Asian physical traits, like black hair, may originate from genetically mutated Denisovans. With this, we've covered the main content of the Neanderthals. Over the past 30 years, human understanding of our origins has constantly evolved. Single origin theory replaced multi-regional theory, but this isn't the final answer. Solving humanity's ancestry mystery fully requires future archaeological discoveries and advances in paleomolecular biology. All right, that concludes our discussion on Neanderthals. Reading notes are below the audio. See you next time!